Hey, good morning, everyone. I'm here with Jacob and Nolan, and we're here to talk about one of the most beautiful examples of evolution in action through this really cool parasitic fungal species called Massospora cicadina. So a little bit more about Massospora. It infects this one type of periodical cicadas that are actually blooming this summer. They come once every 17 or 13 years. It's very interesting. So it's actually in the phyla of Zoophagomycota, which is under one of the ones we learned about before from formerly known as Zygomycota. And the subphyla is also called Entomopathoromycota. It is known as an entomopathogenic fungi, which means it kills and infects insects and arthropods. And one really interesting thing about it is that it actually replaces the host abdomen with conidiospores, which will be very interesting in how it continues its life cycle.
All right, so cicada life cycle. Broods emerge at once and begin reproductive processes. Males will begin to make noise and initiate reproduction. The females will then drill holes into trees where they can lay their eggs. Females can lay up to 16 or to 600 eggs, and nymphs will then fall to the ground where they burrow. Nymphs will then stay underground for up to 17 years. They use tree roots to absorb nutrients. The nymphs will then crawl out of the ground, and then they'll crawl up trees to shed their skeleton. After shedding, the cicadas' wings will fully develop within 6 days. Once the cicada is fully developed, it rushes to mate with other cicadas. The male cicadas use an organ called tymbals to attract females. The females will then shake their wings in response. So Massospora cicadina life cycle. It contains two different stages. The first being transfer of spores through other cicadas, and the second is the dispersal of spores via underground cicada nymphs. So stage one is primarily sexually transmitted. Cicada nymphs accumulate spores from the soil. The spores begin to replace the abdomen and create haploid conidiospores within the abdomen. Once infected, the cicadas will try to mate with the other cicadas. The fungus alters the male cicada's behavior so that they attract both male and females for maximum spore dispersal. White spores will become visible once the back end of the cicada falls off. Exposed spores can transfer to other cicadas through wind deposition. Stage 2 does not affect the host behavior and produces diploid spores instead. These spores evolve to live longer underground, and the spores infect other cicadas through dispersal in the air.
All right. So one of the most interesting things I think about this whole story of evolution and parasitism in this fungi is how do these similarly related molecules become through evolution to be used in all these different ways? And we'll get into all the different theories and how that happens. But one of the most interesting things in the synthetic process is that scientists actually don't know the exact biosynthetic route for how this species of fungi produces these molecules. One of the studies looked at there's several gaps in some of the genes that are responsible for the enzymes and converting the entire chemical cascade for them, one of which being this enzyme called phenylalanine ammonia lyase. So starting from the amino acid phenylalanine, these fungi convert that down the path to cathinones, which is like a similar amphetamine-like related structure. It was actually found in bath salts people use recreationally, which is very, very dangerous. And also the amino acid tryptophan, which was converted into psilocybin through a process as well. You guys might recognize psilocybin from a few other different species, and we'll talk as well as to how these things relate, which is very interesting. So some of the proposed theories as to how and why these fungi produce these go anywhere from, hey, maybe it's not the fungi. Maybe the cicadas are producing them in response to the infection because we only see it in the infected individuals, not the non-infected. Or hey, maybe this is a crosstalk interaction with some of the microbial environment that's already there within the cicadas in the dirt. That could be other types of fungi or other bacteria that's interacting with their secondary metabolites to assist in this chemical cascade of creating these neurochemicals. Or maybe another theory is horizontal gene transfer is being played here. This is something that fungi also do. It's very similar to bacteria and viruses as well. But the theory behind this is that endophytes, fungal endophytes, instead of certain plant species, also produce similar alkaloids. So maybe that some cicadas that also coincidentally eat these species of plants may have led to these genes being transferred horizontally, which means without vertical recombination through sexual reproduction. And the fourth theory we can talk about, we're going to get deep into, is actually convergent evolution, which is fascinating in its own way. So I think the answer is not exactly one of these for sure. Maybe it's a combination, or maybe we haven't discovered it yet. This is why we really need more fungal research. So the most interesting thing about horizontal gene transfer is that pretty much the whole theory is that whatever the organism is, it collects DNA from the environment and incorporates it with their own. So the two main phyla we talked about in class, Ascomycota, Basidiomycota, they have species that produce psilocybin. You have Psilocybes, Conoscibes, and these ones are typically more saprophytes. They can, decay wood, they can be in dung. And some of the theorized roles of psilocybin is that like in these molecules is that like it's used for competition against other, insects that are trying to eat the mushroom, also herbivores that might be trying to eat the host. And also you talked about even interacting with serotonin-like receptors and Drosophila affecting feeding patterns. So it's kind of a evolutionary defense chemical. And now all the way over here across this phylogenetic tree, we have the formerly known as Zygomycota, which we found our Zoophagomycota in our special Massospora. And it's very interesting because horizontal gene transfer might be the link, the bridge, the gap in between these two distantly related fungi into how they both use these compounds in their own niche lifestyle, comparison to parasites versus saprophytes.
All right, so the fourth explanation that Tommy put forward for us was convergent evolution. And convergent evolution is sort of an interesting thing in that it always seems far-fetched to say that these two things evolved in exactly the same way, but it continues to happen over and over and over again. And the reason for this is because all these different environments are putting the same environmental pressures onto different species. And even though the species may be wildly distinct genetically, since they're experiencing the same environmental pressures, they're going to have to adapt to them in similar ways. So a lot of times, just the best strategy to beat a bad situation is going to continue to be the best strategy, no matter what species you are. So what I'm going to do first is I'm going to show you a couple examples of some other species that use biochemical means of controlling a host as a parasite. So firstly, we've talked about this one, is Ophiocordyceps. It's your zombie and fungus. And much like Massospora, is an obligate insect parasite, and it hijacks the insect's behavior in order to spread its spores more effectively. The usage of neurochemicals to to cause abnormal insect behavior shows that this is a viable strategy. This is being done in other places in the wild, which suggests that convergent evolution may be a way in which Massospora came about having these different chemicals. A couple other examples of this that are even further, even more genetically distinct from Massospora are first this guy, Polymorphus paradoxus. This is a worm, so not even a fungus, and it infects crustaceans, which are arthropods just like ants and just like cicadas. And what it does is it causes these crustaceans, which often avoid light because it gets them eaten by ducks, to go up and latch onto rocks near the surface of the water that they live on, which causes them to be eaten by ducks, which is what the worm wants. It reproduces in duck guts. Now, an interesting thing about this is that this is actually the mating behavior of these crustaceans. So it's doing is it's causing maybe a spike in serotonin, it's theorized, to cause these crustaceans to go into a mating frenzy and then get eaten by ducks. Another example is Hymenopepimyces argyrophaga, which is a parasitic wasp, actually, which if you see that little spider there next to that sac and the wasp in the middle image, what it does is it stings the stings the spider, which stuns it, and then it lays an egg on the spider. Long story short, the larva grows up on the spider and then eventually gets inside of the spider's exoskeleton and injects it with a chemical that will eventually kill it. But before it does that, it cocoons the larva in spider web, which forms the cocoon in which the larva becomes a full-fledged wasp. Now, another example of different conversion evolution strategies is actually more related to the replacing of the abdomen that Massospora does with Cymothoa exigua. You guys might have heard of this one. This is a parasitic aquatic crustacean that attaches to fish mouths and then severs the tongue's blood supply, after which the tongue basically rots out of the fish's head, and then the parasite actually replaces the tongue. The host continues to survive, albeit it's not doing as well. But the fish or the parasite stays in there and replaces its tongue. Yeah, there you go. Now, this is a similar niche in the fact that it's literally just replacing the organs of its host, much like Massospora replaces the back of the thorax. The pressure that's causing this to come about is that complex macroscopic organisms are highly reliant on specialized organs and cannot adapt to not use them or to get rid of them. Much like the cicada can't get rid of the back half of its body, the fish needs a tongue and thus can't evolve to get rid of this parasite, because then it's just evolving to not have a tongue anymore. Another example of this is Sacculina carcini. Sacculina carcini attaches to the cloaca of crabs, and then it basically chemically castrates them, and then sort of fills in where those reproductive organs would have been on female crabs, and then hijacks the crab's instincts to care for young and makes it care for its spores, basically. A lot of the same behaviors that are beneficial to crab young are beneficial to these spores. And as a result of that, it's sort of hijacking hijacking an organ and using it in the way that the crab would have quote unquote like to have been using it, but for the fungus's purpose. Or sorry, this isn't a fungus, it's a barnacle, for the barnacle's purposes instead of the crabs. All right, these are our sources. Thank you guys for your time, and we hope we could show you something new.